I was a student in Australia, I went to a conference and I saw the talk by somebody who now became a Nobel laureate and determined the structure of a ribosome. I remember it took her whole career, her whole life, to look at the high-resolution structure of the ribosome. That was before the cryo-EM wave came in and technology came in. Now we can look at big structures like the ribosome in a very short amount of time. So what this person did in her whole lifetime can now be done in a year or a fraction of a year. When you have a microscope of this caliber, then that allows you to look at all sorts of molecules. So we can actually see molecules at an atomic level. So you can see everything that's happening in the cell. In particular, we look at proteins such as the spike protein that is attached to the coronavirus and that allows entry into the cells. This microscope you can use for any protein. So a protein involved, for example, that I've been studying in my lab that looks at the heart diseases, that's involved in making very strong connections to the heart, which is the #1 of cause of death in the nation and worldwide. And also you can look at cancer, for example, and many diseases. It's not restricted to anything. So it allows you to look at a protein at atomic level to then understand how it works, then to create, for example, an inhibitor that stops it from doing bad things to your body and cure diseases. We have been traveling to New York or to North Carolina or to Tallahassee or to California or to Japan, or we've been shipping samples to England because these microscopes are so highly sought after. Everybody wants to use a microscope. They run 24/7. They run every single day, and every single day that's not used is like a huge experiment lost. Now we have this here at our doorstep, so now we can use it every single day, 7 days a week, every single day of the year. That will make an incredible impact to our science and to science of everybody who wants to use it. And it's going to foster a lot of collaborations in different fields coming together to use this microscope. We have an incredible graduate school, and this will allow graduate students and interns and postdoctoral fellows to do research that they couldn't only have dreamed about doing before this microscope came on campus. It's not like a photograph where you take a picture and then you have it. You have to take millions of pictures to then combine them in a big computer and calculate the structure three-dimensionally. This is not something you do in a day. It takes a lot of effort. But once you see that you have an electron density that you can see, you can smell it. It's like, I have this saying in structural biology that you can smell the structure, and then it draws you into, and you're just born as a structural biologist. The first time you see this, you're like, oh my gosh, I'm seeing this, and no one in the whole world has ever seen this molecule at atomic level, just us for the first time. And you get so excited, you cannot wait to finish the structure and show and share it with the world. So like, look, this is how it works. This now everything else, the whole biology of the whole cell biology falls into place, the whole biochemistry. You can explain that, you can understand that, because suddenly like, oh my gosh, there it is. That's how it works. It's like an aha moment, and you work 24/7. You just cannot let go and cannot wait to share it with everybody.